Terrific. Thank you all for being here. I'm Leslie Subak. I'm the chair of obstetrics and gynecology, and I have the honor of moderating this session. Our first speaker is Joachim Hallmayer, who is a professor in the Department of Psychiatry and Behavioral Sciences, and he will be presenting a talk, Autism Spectrum Disorder from Genes to Neurons.
Thank you. Thanks so much for inviting me. Thank you very much. I'm going to give you really a big overview, and the reason I'm doing it is two things. First of all, 10 minutes are very short to go into too many details, but also very simple. The grant basically we got from MCHRI was one of these new ideas programs award. Basically what this was, it was a grant, and you won't believe it, but it gave us a lot of money to meet, not a lot actually, a little bit of money to meet, and we supplemented it with lots of wine and got together and discussed basically how could we tackle the overlap between autism and epilepsy. And what I'm describing you today is basically a little bit of some of the things which came out of it, not so much in terms of results, but in terms basically of projects which we started through these groups. The title, the overlap between autism and epilepsy was, if you want to speak, the starting point, but there's a lot of discussion which came out, so the programs of what we are doing right now is a little bit different from what we originally imagined, but it really started basically out of this, this getting together over wine and discussing it with a couple of people from psychiatry, myself, Ruth O'Hara, Greg Garner, Antonio Harden, neurology, Brenda Porter, John Huguenot, Joseph Pavisi, material science, Nick Melosh, and in psychology, Antonius Nocia. So, but basically to understand basically where we started, the starting point for this was really autism spectrum disorders. What are autism spectrum disorders? And there were many talks about it, so I don't want to go in any details, but basically at the heart of it are impairments in social communication and interaction. The important thing is that it's basically entirely behaviorally defined. Okay, so there is no marker, nothing. So it's entirely basically based on your judgment, so to speak. Now, the etiology of this disorder of this group of disorders is entirely unknown, and the therapy is symptomatic. And I spent basically my entire career from 1989 onwards till a couple of years ago to figuring out the genetics of it. And in a nutshell, basically, years and years of work where it stands right now is that there are about 10 to 30% of children where probably rare variants contribute significantly to the development of autism. Now the interesting thing, and that's why we got together, is that these genes basically overlap with epilepsy. And the things we mainly discussed then is basically how can we explain this overlap. About somewhere between 10 to 30% of children with autism develop epilepsy. Many of them during puberty, basically, later on. We are not talking about early onset epilepsy, but many of them later epilepsy. And the question is basically, what can this tell us about the functioning of the brain? The remaining of the 85% is either common alleles or environmental factors. And we can now discuss till the cows come home how strong is the genetics and how strong is the environment. I think it's a ludicrous discussion in a certain way. Both basically play a role and both interact. So what do we know about these mutations? And that's important to memorize, I think. It's important to point out. Few, if any, in my opinion, none of them is actually specific to autism, but we can discuss. I mean, I'm often wrong, and I might be wrong in this one. There's definitely a huge variation in severity in carriers. Few are associated with specific clinical subtypes, and we have tried this over and over and over again, basically, to put subtypes towards some of these mutations. Some of them we can have some ideas, most of them we have basically no ideas. The most are also found in healthy individuals, and the more we sequence, basically, the more we find mutations which have been claimed to be specific to autism than in a healthy individual or an individual with ADHD or with pure epilepsy, you name it. And then there's a huge overlap with intellectual disability. Now, is there light at the end of the tunnel? So basically, that's where this all started. So basically, what we figured out is that these mutations are not random. They converge on a limited number of pathways. And some of these pathways are peripheral, like the immune system, but the most important pathways where they really converge on are basically into the presynaptic functioning and the postsynaptic functioning of the neurons. There are basically, if you look at these genes which have been implicated, and again, we could discuss which ones are true ones and which ones are positive. Whatever it is, they are really converging on a small number, basically, of different pathways. As you know, then came the time of the induced pluripotent stem cells, and this got us very excited. And again, life is always very, how should I say, accidental. When I was shopping basically at Whole Foods, this is no advertisement for any shop or something in Redwood City. I bumped into Ricardo Dolmetsch, and we basically discussed this literally while his wife was waiting, and had this great idea that this is something we could basically move forward. And then we applied basically for a grant to use this technology, which allows us basically to get neurons. Before this was even published by Yanamaka, for a season of several reasons we knew about it, and so on and so on and so on. Then we got the grant back, and it was said, yeah, that's a great idea, but we are the wrong persons to do this, which is always encouraging. Having said this, basically, we moved then forward, basically, and we started out with a proof of principle. And the proof of principle was basically, and this was, think a little bit back, so now we know much more, but basically our question was very simple, actually. Is the effect of the mutation represented in the functional properties of the neurons derived from the peripheral cell? Because that's the first question we want to ask. The first question basically we want to ask is, can we capture anything in these neurons or not? So what we basically did then is that we started to collect these single gene mutations. And I think we have, in terms of iPSCs, probably meanwhile one of the largest collections, if you want, so of iPSC lines from individuals with different deletions and mutations. And this is a growing number. From individuals which have been very well assessed with autism. We then basically started to derive iPSCs and neurons from them, and this is for 22q11 deletion syndrome, but we have done many others meanwhile, and many people in the field have done this. So basically what all of these basically show is that yes, the effect of the mutation is represented in the functional property of this derived from this peripheral cell. So basically, yes, we can recapitulate at least something of the pathophysiology of these cells. So, the next question is, and what about this common or idiopathic, non-syndromic, whatever you want to call it, about this common? I mean, it's nice that we can recapitulate, let's say, a mutation which is in a calcium receptor or in a calcium channel or whatever it is. But the big question is basically, if this is all we can do, then it would be a little bit disappointing. So the question basically, and that's what we discussed with this group, or that's why we got together, is basically how can we move this forward, basically? How can we get to the idiopathic phenotype? Is there a convergence of this phenotype, at least in a subgroup? I.e., can we use this information which we have derived from the single gene disorders then to move it forward to at least subdivide or to at least find some pathophysiological convergence in this group of idiopathic autism? And now I'm talking more about the future because it's all ongoing. So there are basically two principal approaches we discussed, which are very obvious, and you might think, yes, you need a lot of mind to come up with this. You don't, it's very simple. There are basically two basic approaches, I think. The one is that you use well-defined subtypes. One subtype is, for example, autistic individuals who develop epilepsy. Another subtype we are looking at is individuals which have larger head circumference. The other possibility, and this is the one I want to talk 2 or 3 minutes about, is very simple. We need a bigger boat, meaning basically we need significantly more samples. If you look at iPSC studies, the maximum number of people have studied are 5 to 6 individuals. So we have to crank this really up. If we want to make this technology, I think, usable, what we have to do is really to crank it up exponentially. Now, what we are trying to do is not go from 1 to thousands, but let's say a little bit bigger, basically. What are the biggest hurdles or what are the challenges? The biggest hurdles are definitely costs. I mean, as you know, even today, to derive an iPSC costs you a couple of $1,000s, depending how you want to do it. So it's very labor intensive. And then you have only the iPSCs, and you have to get your neurons. That's another $3,000 to $5,000, whatever technology you're using. The next one is availability of samples, and then there is in-depth phenotyping on large samples, which is very important in psychiatry, at least in psychiatric disorders. How many minutes? Oh, 5 minutes. Gosh, I'm fast. Good. So, along came basically what's called the Induced Pluripotent Stem Cell iPSC initiative, and for which reason ever, we were basically selected one of the sites, as one of the sites to collect specifically individuals from idiopathic autism. The California Institute for Regenerative Medicine did not want any samples with a known etiology, interestingly. So they basically, already from the beginning, they said, we don't want any known etiology. So basically, what they paid for is relatively simple. They paid for us to collect the sample, to characterize the sample with a reasonably, or with I think a very, not totally deep, but it's a very well-defined phenotype with the standard assessments. And they basically provided us with iPSCs. So basically, in the last years, what we have done then is is basically to collect a really, really large sample, or really large sample is exaggerated, but a sample of over 300 individuals with autism and controls, which we now basically have the iPSC cells on. And as a next step, we got just started now a grant basically, which we got funded by NIMH after several submissions. Again, an outpour of this discussion we're having. So this CHRI funding really, I think, paid off, there's no doubt. So basically what we got funded and what we are starting to do is that we want to take 100 cases and 100 controls. I know that's not exactly what we need. I know that's not exponential, but it's significantly more what has been done to date. We want to grow them basically up. We are partnering with a company to grow them up. We partner with a company because we wanted a technology which is replicable. For this type of methodology, I think academia can be very, has advantages and disadvantages, but it can be very tricky to set something up which you have to do over and over and over and over and over do again. Reminds me always when the big sequencing projects came out, and I was at one of the Hugo meetings, and somebody then got up and asked one of the speakers, I don't know who it was, one of the very famous speakers, whether this project basically he proposes means that now hundreds of postdocs will be repetitively sequencing individuals. And I think that's a little bit similar here. You cannot, I don't think you can tell somebody grow up 100 or 200 of the cell lines. So I think it has to be done somehow in a commercial entity. We want to grow them up, or we are growing them up basically to 3 time points, and then we do the usual stuff, the gene expression, and then we want to relate basically this to the group of the dysregulated genes. We are doing a number of neuronal assays, and that's what we are working currently on, basically to develop methods, again with another company, together to automatically measure then things like neurite length, number of branch points, overall complexity, and then we want to do also calcium imaging. It's now a method available where we can do the calcium imaging in the live cells, fortunately, so, and at 3 different time points. Last but not least, basically, the goal is then to relate these gene expression profiles and the neural to the behavioral phenotypes. Again, it is a very ambitious project on the other hand, something I think which has to be done, which we want to do. And I'm talking now, but there are tons of people basically which worked on these projects, and these are just a small number of them. But I definitely want to thank them. And last but not least, I want to thank all the families for the participations and all the funding. I mean, as I said, the Child Research Institute, where it got off, and then NIH, CIRM, the Science Foundation, Phelan-McDermott Syndrome Foundation, and others basically have funded our projects over the years. And I want to thank especially CHRI again for starting basically the whole thing. Thank you very much.
We actually have a little extra time for questions because we're running a bit ahead of schedule. So, any questions?
I was too fast?
Yeah. You want to do it again and talk slower? Okay.
I could talk slower.
Or add some extra good stuff.
So,
okay. Yes, please.
So that was wonderful. A question. I have 2 questions. One, how much variability is there between IPS lines versus the mutation? Because that would determine how sensitive you're in terms of numbers. And then the big question in the autism field is how much is immune mediated and how much is cell autonomous in the neurons. And so can you give us sort of a ballpark estimate about all those variabilities?
I cannot even give you a ballpark estimate. It's something which we are trying to do right now. One of the things is, unfortunately, because of time reasons, I threw out the picture of the different variances you have to take into account. And there are very, very many different points. The first point is basically the place where you take the sample from, whether it's peripheral blood, and so on and so on. And then you start on, then basically, so each one of these points has its own variability. So the variability is basically is quite enormous. So in order to address this, what we are doing is that we have about 40 samples, which we will do, where we have 2 derivations each. I mean, 2 derivations from the beginning. I mean, not usually people, what they do is that they derive in 3 different lines or something. But this is previous, so basically we do the whole experiments basically 40 times over again, so to speak, in order to estimate then this variance, which is, I haven't found any good estimate of this at all.
Right.
Your second question, that's actually a very good question, and that's a pure gamble. In this study, we are looking at neurons, so we are looking what is cell autonomous.
Right.
You could also go, and Sergio Pasca is doing this at length, you could go to these 3D cultures. We had long discussions. I mean, the problems of the 3D cultures is that you introduce a whole other range of variability. So unfortunately, I can't tell you. The idea of our study is that there is something which we can discover which is cell-autonomous. Whether this has something to do with the disorder, that's then the second question. But we study a model, okay?
But in a complex genetic system, each of the iPS cells has a different genetic background on top of the disease allele, right? So if you're having each disease allele have a small effect, it seems like that might be a challenge for determining the variability, the particular function.
Oh, absolutely. This is exactly the challenge. That's why I said we need a bigger boat, because the only way to address it is that we have large numbers in order that there will be some subtypes which will be hopefully falling out. I agree entirely. It's an entire gamble. We might do 10,000 cell lines and find nothing. We might find, I don't know. Yeah. If I would know the answer,
But it's certainly an answer worth trying.
I think it's an answer worth trying, and we are not totally blind, because I didn't have time to. We think that there are some lessons we can learn from this single gene mutation, so that we are not searching. This sounds like totally blind. There are some things which you do totally blind, but there are others which are really hypothesis-driven based on these results. I just don't have time to show these results. So it's a mixture of both. Yeah. Yes, it is a risk. That's why it was so important to get this initial funding, you know, because we don't know, but it's, you know.
Right.
I mean, other questions?
Okay, I have a question that's going to back up from science, and that may be of benefit to a lot of people in the room. I was really intrigued by your comment that you got back on a grant review. We are the wrong persons to do this. Lord knows I've gotten that comment before. How did you go about assembling your multidisciplinary team? How did you go about finding your collaborators, and what value has that brought to your science?
I mean, as I said, I think that's where this initial funding of MCHRI was so important, because it brought together, let's say, people from basic science as well as people who are more clinical. I'm more clinically oriented, so I'm not the person who knows all the details about the neurons. I mean, God forbid. Though, yes, so this collaboration is the most important. And the most important is that people talk to each other and understand, I think, the limitations of both sides. I mean, there are huge limitations on the clinical side, but there are also huge limitations, I think, on the basic science side, what can be done. And this is absolutely important. Now, how do we find, mainly just by going out and asking people, I mean, you know.
Or shopping at Whole Foods.
Shopping at Whole Foods, for example, exactly. That's one possibility, shopping at Whole Foods and being lucky that somebody likes the same food as you do. No.
Great. Thank you very much.